I would like to thank Professor Williman and the organizers of MyBlueCosmET'IC for inviting me to speak today. The title of my presentation is Human Skin Bioprinting to Transform Innovation in Skincare Research. We set up CTIBIOTECH to help researchers worldwide in their areas of pharmaceutical and cosmetics research. We have an international research and development program working with companies, but also working with universities and hospitals to advance skin and pharmaceutical research of the future. Our work is designed to increase the development of dermatocosmetic skin products of the future. We have ex vivo models on skin biopsies, complex multicellular in vitro models, and we are pioneering bioprinting of skin by tissue engineering. We have developed ex vivo skin models where we take donations from local hospitals and make them available for testing active ingredients or finished products. We can use these to test the effects of UV irradiation and sunlight, but also to look at the effect of active ingredients on the color and strength of the skin, and also look at the histology of the skin to understand the effects deep into the dermis. Our hair models have been routinely designed to look at different sections of the hair, from the dermal papilla to the color content of the hair and the aging of the hair, to really understand if an active ingredient will have a direct effect on the hair follicle and its lifespan. Some years ago, we created CTI Baby Skin, the world's first pediatric skin model to evaluate safety and efficacy of active ingredients and formulated products. This ethical skin model has an active immune system and can be used to test any product which comes in contact with a baby's skin, including food, cosmetics, baby powders, creams, or anything else which could cause irritation to the baby's skin can be evaluated in this model. With the help of the French government, we have created one of the most advanced 3D bioprinting platforms available to dermatocosmetics researchers across the world. The idea behind 3D bioprinting is to create reproducible, effective, and personalized skin models, which allow people to test many combinations of active ingredients on skin. To explain how we do this, we take a very small donated sample of skin from the hospital, we pull it apart and disaggregate all the cells, allowing us to grow them in vitro, and then to reprint them into a full thickness skin model, which can even represent the sinusoidal shape of normal skin and expressing the normal markers that one would expect inside skin. And we can then use this for testing of any product which is destined to be placed onto the skin. Using advanced software, we can design the thickness and the shape of the skin, and we can also decide which cells will go into the skin, from the cornified epidermis right through into the dermal or hypodermal areas. We also spent a lot of time to design the culture medium and indeed the bioinks necessary to grow healthy skin tissue. In this live video, you can see a 3D skin model, full skin sized, being printed. The printing itself is relatively simple, thanks to the bioprinters which are now commercially available from companies including Cellink in Sweden. However, the hardest part of technology like this is to make sure that the cells are happy within the bioink so that they grow into a proper full-sized skin model. In only an afternoon, it is possible to print 100 or more models of reconstructed human skin using this technology, which means in the future we can test more variants of active ingredients and make much more active cosmetics for people to use. Manually created 3D skin models have been around for many years, but more complex models are still needed for research and product development. We have started with our 3D bioprinting platform to create much more complex models which contain different elements of the immune system. In order to create 3D immunized skin models, we have our standard 3D bioprinting together with the normal epidermis and dermis format. But we can then take blood or bone marrow from a donor, and we can separate specific immune cells to add into the skin, which allows us to modulate the skin formulation and gives different skin pathologies an opportunity to be mimicked in the lab. In our collaboration with the BASF Corporation, we have been developing macrophage models, looking at the M1 system and the M2 system in order to be able to test if active ingredients have an anti-inflammatory or a pro-inflammatory effect on the skin. These models are very sensitive and respond very well to challenge. When challenging these models, they have a very good response in terms of cytokine release, depending on the challenge that has been presented to the model. Using a CBA spread analysis from Becton Dickinson, we can measure many different cytokines being produced from the skin model in response to the induction or treatment. Taking our skin models a step further, we have started to develop very sophisticated modeling of the sebaceous glands inside the skin. Sebaceous glands of the skin are extremely important to regulate skin homeostasis in terms of lipid production, thermoregulation, photoprotection, antimicrobial activity, pro and indeed anti-inflammatory properties, and many other roles. Already in our laboratory, we have some of the world's most advanced sebaceous gland models, from developing sebocytes from a wide range of patients of all different ages, through to looking at sebaceous glands in situ in ex vivo skin, and also extracted sebaceous glands in ex vivo culture. We also have clinical models that we are developing with our partners in hospitals as we move towards clinical trials. Together with the BASF Corporation, we have developed the world's first human 3D ex vivo sebaceous gland model, which allows people to look directly on the sebaceous gland to understand if the product has an effect inside and outside the gland, looking at the lipid production and the overall growth of the gland with regard to the active ingredient. Building on the 3D ex vivo sebaceous gland models, we took this a step further to do 3D bioprinting of sebaceous glands in vitro by taking skin samples from patients and creating same donor fibroblast keratinocytes and sebocytes, bioprinting them into 3D models to be able to screen a wider range of products aimed at regulating or changing the components of sebaceous gland production. The printing of microsebaceous glands was achieved, and we can see actually the individual glands after they've been stained with Nile red. You can see here a solar system of donor-specific human sebaceous glands being produced inside the 3D skin model. And this model can now be used to test active ingredients and final cosmetics destined for skin. We have taken our models and we have subjected them to rigorous confocal microscopy, labeling the glands for the typical markers, vimentin, mucin 1, keratin 7, to look at the structure and also to try to understand the active areas of the gland and how they develop and how long the model must be matured before we can then test an active ingredient onto the model. Using our microsebaceous gland 3D printed models, we challenge them with testosterone, linoleic acid, finasteride, and TOFA combinations, showing that the model was highly functional, with different ranges of oils being produced, including differences in squalene production, showing that we have a very good model for testing active ingredients of the future. Our 3D bioprinted skin models are designed to be flexible, so that we can change the components within them to model different skin pathologies and different phototypes. In the future, we hope to be able to create a wide range of models useful to both the research and to product development industries. Not only do we have the ability now to print basic skin models for toxicology screening, but also models with an active immune system. Under development right now, we have microbiome models and also sensitive skin models with a neural base. This allows us in the future to be able to modulate the models according to the end research requirements. One of our newest projects is called the LipoCosm Initiative. This is designed to bring private companies and public organizations together to help advance all the different technologies required to have active ingredients of the future useful for the cosmetics industry. We're proud of this project because it allows us to bridge the gap between the public and the private organizations, and we believe that this will help the future of cosmetics development. So with that, I would like to thank the organizers of MyBlueCosmET'IC 2021 for inviting us to speak, and also to thank our team at CTIBIOTECH, and indeed the teams at BASF, Beiersdorf and Novatech for all of the help that they have provided with us in these projects. Thank you very much.